This is a production of Cornell University.
And thank you for joining us again. As a reminder, my name is Bahia Zal, and if you attended the previous talks, maybe you remember I'm a second-year PhD student in plant breeding and genetics here at Cornell University, studying hemp with Dr. Larry Smart. Just so you know, these webinars are being recorded, so you're able to return to them, rewatch them, or share them with others. They're hosted on our Cornell School of Integrative Plant Science YouTube channel. Also, you can find more hemp resources on our Cornell Hemp website. I'll include those links in the chat if you want to check them out. As a reminder, as I said earlier, if you have questions for the speaker, please put those in the Q&A box, and we'll answer those at the end. And then any other comments you can put in the chat box. So thank you for joining us. I'd like to welcome our speaker today, David Gang. Dr. Gang is a professor in the Institute of Biological Chemistry at Washington State University, and he's the director of Washington State's Center for Cannabis Policy Research and Outreach. He has special expertise on plant medicinal compound identification and production. His lab uses many different approaches, such as genomics, proteomics, and metabolomics to investigate metabolism in plants and other organisms. He received his PhD in plant physiology and biochemistry from Washington State University. He's also served as the president of the Phytochemical Society of North America and as a chief science officer for a startup company, Botanisol. He's dedicated to improving the life of people by increasing food availability and nutrition, while also reducing production costs by better understanding plant bioactive compound production and how plants interact with their environment. So thank you, David, for joining us today. I'll let you take it away with your presentation.
Okay, good. Thank you again for the invitation to come and speak in this seminar series. I'm very excited about the opportunity. Today, I'm going to be talking about our efforts to try to integrate hemp research in Washington State University. A number of years ago, I got involved with a project with some individuals at the University of Minnesota looking at how cannabis produces bioactive compounds like the cannabinoids. We did one of the first projects looking at transcriptome of the trichomes on cannabis sativa. And at that time, you couldn't work on hemp in the United States. You really couldn't work on cannabis in the United States. Only a few people could. And my expertise in trichome isolation and metabolic pathway characterization was part of that effort. And that got me interested in looking at the plant. And I decided back in, it was like 2006, a long time ago, that there really wasn't a lot we could actually do with it at the time because the regulatory situation was such in the United States that it was basically impossible. But things changed over the years. I moved to Washington State University. And once I got here, it became clear that there was a big effort at Washington State to look at cannabis, but there really wasn't any effort to work on hemp because, again, there was not an opportunity to do so. Years have passed. We had a couple of farm bills passed, 2014 Farm Bill. Washington State did a very good job of trying to discourage anybody from working on it. Other states, like I know New York and Oregon and Colorado especially were able to get these pilot programs going for hemp research. We weren't able to do that. But when the 2018 Farm Bill came along, that opened up the door so we could finally move into this space. And I've been very appreciative of the efforts and collaborations that have been established across the nation to try to move hemp research forward. That's allowed us to do things that we really couldn't do even a decade ago. And it's opening up doors for everybody across the nation, and I think worldwide, to be able to work on this very interesting plant. One of the things that became clear to us in Washington State early on was that if we're going to work on hemp, as we're moving to more the industrial hemp side, because I believe that fiber is really going to be the future of hemp. Yes, there's an important role for cannabinoid hemp, CBD hemp, chemical hemp, whatever you call it, or essential oil hemp. That is an important part of working with hemp, but there is a limit to the scale of what that industry is going to be. Fiber, on the other hand, has a great potential to really do a lot to replace petroleum the humankind's addiction to petroleum on this planet. And that's one of the things that I'm very interested in trying to promote. One of the ways that we do this is we work with partners in the area. Here in this picture are some of our partners from the Yakima Nation. They have joined with us to try to understand how hemp can grow and be developed as an important commodity in our region, and how they, as a community, can be involved with this and promote its use in a way that will benefit their community especially. And in order to do this, some of the things that we have to deal with is there's compliance issues with state and federal law. But in our area especially, and those compliance issues are relatively easy to deal with now at this point. But the bigger issues for us are really trying to optimize varieties that work well in our area, figure out what the best production methods are for our area. Dealing with weed management is an issue as well. And especially, we need to be able to establish processing facilities here in the U.S. At the multi-state meeting a year ago in Colorado, we had an individual come and give a presentation and talking about how the development of these processing facilities is being promoted and developed in the nation. And 5 years ago, there was one processing plant in the U.S. As of a year ago, there were over 20. We're getting close to 30 now. It's great to see that happen, but there's none of those in the Northwest still. And we're very interested in trying to figure out how we can make that happen and move them into our area. And in order for that to happen, we have to demonstrate, of course, that the plant grows well. And as you can see, looking at the plants growing behind us here, if you're not careful, the plant grows too big, actually. It grows exceptionally well in Washington State. We're kind of like an ideal environment for it. So we're super excited about being able to work in a way to find partnerships and develop ways that we can promote this plant and its development and the economy based on its development in our region. And as I like to talk about it, we think about hemp in a global, local level, where we think about it to function locally, to produce it locally, to process it locally, produce products locally. But we do that as a global effort where everybody nationally and internationally can promote it simultaneously, so that products that include hemp are accepted and actually desired by everyone across the planet. And that will build our local industries very well. So here are some of as I mentioned, it's a great location to grow hemp. Washington State actually has some of the largest diversity of environments in the nation. But here are some questions that we have. What types of cultivars work well in our area, and especially in different environments across the state? We have a rainforest to a desert in our state and everything in between. What are the suitable locations, and what types and varieties grow in those locations? And which of those conditions are really the best for different types of hemp? And then how do we grow different types of hemp in the environments that we have in a way that leads to the best product quality and that also follows the regulatory compliance issues that we have to deal with? Finally, thinking about all of that, really, what are the best hemp products that are the best suited for Washington State? And how do we make hemp available for those products? And that's going to include breeding efforts. So we have started moving in that direction. I am by no means a plant breeder, as was mentioned by Bahia. My training is in plant biochemistry, but I'm working with individuals that are, and we're trying to move this effort forward in our area in that direction. Also, the way that we're doing this, as I mentioned, we have partnerships, and that's really the way that it works. If you're going to build an industry, you have to build the right partnerships. Big partnership is with the Industrial Hemp Association of Washington. IHEMPAWA is the abbreviation for that. And many growers in the area, especially our tribal partners. As I mentioned, the Yakima Nation, the Confederated Tribes of the Colville Reservation, Tulalip Tribe, Puyallup Tribe, and Nez Perce Tribe are major partners with us in the area. They're all very interested in developing hemp for its fiber and grain applications. And we're very excited to work with them. They're partners with us on several grants that I'll talk about in just a second as well. We also have a major partnership with Oregon State University and UC Davis, a West Coast tri-state partnership to try to develop a hemp bioeconomy on the West Coast. And I'll talk about that briefly a little bit later too. We also have partnerships with ARS, and part of the title of my talk is talking about this. A little bit later, I will talk about what we are doing to contribute to this effort for the germplasm collection. And of course, the leaders on that are Zach and Tyler, and I definitely recommend you talk to them for details. I'm just going to give you, for those that are not familiar with this system, I'll give you a brief overview of what it is and how it functions, and talk briefly about what we're doing to help contribute to that. And I think it's a great effort and an important effort for us to be involved with that. And finally, we're involved in many variety trials that are helping us both in our effort to figure out what grows in our area, but also kind of at the national effort to see what grows in different parts of the country. There are varieties that some people love that perform exceptionally well in the Upper Plains area. Many of you may know of it. It's called X59 for a grain. It grows really well in eastern Montana and the Dakotas, and it is not a very good variety for our area. We've had many people try to grow it, and it just doesn't work. We don't know why it doesn't work so well in Washington and Oregon, but there are others that perform a lot better. And that's interesting and useful information to know. And why is it that different varieties perform in different areas differently? That's important information for us to know as we're trying to build this industry so that, as I said, we have this global local economy that we're developing. Okay, in order to be able to do this, we've been able to get some very important grant support. The director of the Hemp Association in Washington, her name's Bonnie Jo Peterson. We and I have been working together as close partners for many years now in trying to develop this since we could first start working with it right after 2018 Farm Bill was passed. It took a year and a half for Washington to get everything lined up so we could actually start working with it. So it wasn't until 2020 that we could really be involved. And at that time, Bonnie Jo and I were talking about, it's like, man, it'd be wonderful if we could get grant support to work on this. And about that time, we talked with Oregon State. And Jeff Steiner's the lead on this grant. It's a NIFA Sustainable Ag Systems Grant, or SAS grant, and it's focusing on the West Coast bioeconomy. This is a very wonderful grant to help us all work towards developing this industry in the Western United States. And a lot can be learned from the efforts and the results of this project for other parts of the country. But 4 years ago, this was like an imagination. Could we do something like this? There's no grant support, there's no programs. But in just a couple of years, that all started to change. We've been very lucky in being able to get access to grant funding that helps us. We got a SAC grant, a Specialty Agricultural Crop Grant or something like that. Oh, Special and Alternative Crop Grant from NIFA. That supports the effort for the germplasm collection development. We got a $45 million Climate Smart Commodities grant with Oregon State, looking at integrating regenerative practices in potato cropping systems, which is very, very hard on the soils. Hemp can help alleviate those problems or repair the soils. And so a part of what we're doing there in that grant is looking at how hemp can help benefit farmers and growers. We just recently got a $700,000 grant for a year to look at pesticide uptake in cannabis plants, including hemp. And I just was announced to us recently that we're getting a grant from the DOE to look at the integration of hemp and biochar in crop rotation systems and what impact the integration of those two components into that cropping system has on both carbon sequestration, but especially carbon durability and greenhouse gas emissions. And also, of course, it's not going to work if you don't benefit the crop. So we're looking at yield and, quality product quality for those crops. But we're excited about this grant. It's a 6-year grant that's going to be starting up. That's starting up this year. We're very excited about the future for that. And we're also, we just heard we're having another grant that's going to be supported that we're going to be able to talk about very soon. So things have changed completely in just about 3 years in terms of the grant scene in the US. We went from thinking maybe someday there'll be grant opportunities to all of a sudden there are many opportunities. And that's very exciting for the hemp industry and for hemp development nationwide. And we're not alone in, being able to receive this kind of support. And as I said, Washington State is an ideal location for the hemp ag industry. Here's our field trial from a couple of years ago at our Prosser Research Station. You can see the plants. This is, I think this is like mid-August. The plants are doing really, really well. This area is surrounded by hop farms and tree fruit, from apples to cherries to pears, and some berries. There's a lot of blueberries in the area as well. And hemp just loves growing in this area. And it's very easy to perform research, but it's also difficult in some other ways. And I'll talk about that in a minute, because some things come along that make a challenge for you. But as you can see, as things mature, the plants develop as you expect. And we're very excited about the opportunity to perform research in this area to help support the industry development. Our variety trial in 2021 was the first one where we actually were able to put out plants on this plastic tarp kind of a system, drip irrigation underneath. We did a density trial where we grew plants at a higher density and at a lower density to evaluate how they perform. We also did a comparison of transplants, as you would expect for the system, but also direct seeding, where we punctured the tarps and put the seeds in to see what we got for a difference. And I'll talk about results of that in just a second. And as the plants grow, You see a big difference at first between those that are transplants and those that are the direct seeded plants. The direct seeded these were started at the same, planted at the same time, and these are a lot further behind. The transplants take off and seem to perform a lot better at first. We get to the end harvest time, and we see different varieties and different plots have different kinds of results, which of course is what you would expect. But what was really interesting, and here's what it looks like in 2023, we actually got a little bit better at this. And I love these pictures at harvest time, how the different plants have their different colors. One of the things we're also interested in looking at, me as a biochemist, is not just cannabinoids and terpenoids, but also other compounds that hemp makes, like some of the flavonoids that lead to these colors. And these kind of variety trials will allow us to do that. But the big thing that we're interested in, of course, is supporting the industry, which is going to be looking at terpenoid and cannabinoid profile. And so we've got students, as you can see here in the background. A couple of students are out there collecting data from our plots. This is how we do it. It's manually intensive, kind of tedious work, but it's the kind of work you have to do to get results using the research scale efforts that we're able to do here on our 3+ acre plots here. And I showed this picture just a second ago. This is that comparison of transplant versus direct seeded. And at first we thought there was going to be, the transplants would outperform the direct seeded, but when we actually look at the data, we see there's actually the opposite happen. We have a significantly higher yield when we direct seed the plants as opposed to put transplants in the ground. This is two different varieties, lifter triploid and diploid from a supplier in Oregon. And what was really interesting to us is it's almost a double increase in yield when you do the direct seeded. We think the reason for this is root architecture. When we pull out the transplanted plants, they tend to have highly branched roots, multiple sort of tap roots, but more like branches. The direct seeded plants tend to have a single tap root with then branches off of that. And we think that single tap root is really important for the plant to be able to support a larger yield. At least that's what it's looking like in our area in Prosser. And we're evaluating data from last year and year before as well to see if this is holding up, but it looks like that's the case. This is really interesting because it does tell you that how you grow the plant really can have an impact on what your results are going to be. All right, we're also involved in the dual grain and fiber, the grain and fiber and dual grain fiber variety trials. This is led by the multi-state project, what's called the S1084 project. There's over a dozen universities nationwide that are involved with this. We grow ours like this in these 10 by 20 foot plots. We found that it's important to have a wide enough plot to get proper height of the plants. If you do a skinny plot like this one here, this is only 5 feet wide. The plants here are about 50% shorter, or maybe they reach about 75% the height of the plants when you put them in a larger plot. And that height difference makes a difference in yield as well. So how you grow the plant really makes a difference. How you study it and how you grow it for your research makes a difference. If you're going to grow it in a large field, ultimately, which is what I think everybody does at the industrial scale, you're not going to grow them in little tiny plots. You're going to grow it at a large acreage. You need to have a plot that is going to be similar in terms of its ability to produce the plant as that high acreage plot would do. And at the same time that we grew these, we grew some other strip plots that were a quarter acre, an eighth of an acre in size. And we saw that we got the same plant height, same plant diameter on average in the middle of the plots is what we got on those larger strip plots, as opposed to these smaller little plots where the plants are really, you have a major edge effect that affects your ability to grow the plants. Now the way that we're growing them, we do have this aisle. You can see that in between the plots. That's important for us to get into them and harvest them. These plants in our area, they get to be 12, 13, 14, 15 feet tall. And if you have these all next to each other, it's like a jungle. It's very difficult to find where anything is. So for us to manage it, this is how we have to do it. But it's interesting, the results that we get. Here's my field manager, Greg Garza. He's 6'4", and you can see the plants really dwarf him. We have two different varieties here on the left and the right, and there's a big difference in height that you can tell here. And we see that when we evaluate these different varieties. All of them perform very well in Washington, but some of them really outperform others. And we're very interested to see how that's going to go forward in our research efforts. I don't have good results from 2023 because unfortunately our plot that was assigned us at the Research and Extension Center had been used by hops growers before, a couple of years before, and they didn't tell anybody and they didn't tell us until after we did some work and our plants weren't surviving. that they had unfortunately sprayed the field with some long-term residual herbicides. And hemp is apparently very susceptible to those. And so we didn't really get any growth from those plants. Each of these plots has close to 3,000 plants that comes up and grows. And I think on the best plot, we had less than 200 that survived. Most of them, we had 2 or 3 or 5 plants that survived this herbicide treatment. What's interesting about that is some of them actually grew okay. And so we think that's interesting, and we're evaluating, looking at we're actually doing some genome sequencing with Hudson Alpha on this to evaluate what the differences are between those different individuals that survived versus other individuals in another area where the same variety was grown, but they're not the ones they're a typical plant from that variety. Very curious to see if we can find information about herbicide resistance that might be useful for others in this effort going forward. Here's what they look like at the end of the season, kind of actually on the opposite side of that plot. There's Greg again and Bonnie Joe, and we see a big difference in maturity date for different varieties and the grain yield and the fiber yield as well. And this effort is an ongoing effort, but we're excited to be part of it to see and to demonstrate to people in our region that hemp can grow very, very well in Washington State. We just need to give it a chance and get people involved. One of the ways that we've done that is get our tribal partners involved. Here is, we had a 12-acre field that we grew 7 different varieties on this past year. And these are fiber and dual crop varieties. Here's where they are right after planting. This is one that's grown mainly for fiber. It's a French variety called Futura 83. We really like this one in Washington State. It grows exceptionally well, produces apparently good bast fiber quality, as well as having very good herd production. And for a lot of the individuals that we're working with, especially the tribal partners, they're very interested in hemp because of the promise it has for helping with the housing problems that they have. We have a housing crisis in Washington State. We're actually the #1 state in the nation for lack of availability of housing, of appropriate housing for people. And the tribal partners especially have a major problem. About 20%, between 10 and 20%, depending on the tribe, of the tribe members don't have housing that's appropriate for them. And it's not that they don't have the ability to have it, it just does not exist. And so they're very interested in finding ways to produce housing in a way that's going to be, #1, sustainable. That's the big thing that they're most important, that's most important to them now, is they need to have a sustainably built house. And something that's going to last a long time, something that's not going to be made out of materials that are toxic to people, something that's not going to promote mold growth, which a lot of our chipboard-based housing in the Northwest tends to do. A lot of the housing in Washington State has been condemned because of black mold growth. It's a big problem in our state. It's one of the reasons we have a housing shortage. Hemp can solve a lot of these problems. You build a house out of hemp, you don't have those problems. You don't have the mold problem. You don't have rodent problem. You don't have insect infestation problems. This has been demonstrated in many parts of the world, and we're very excited about being able to integrate this type of housing into our area. The tribes are very excited about this, so they're very interested in what they can do to help support the development of the industry and be part of this and build up the industry within their communities. So this is the field at the end of the season. This is what it looks like. We're not quite at harvest time. We could be at harvest time. They harvest a little bit later than near. This is Bonnie Joe again and our partner Jackie. And as you can see, the plants, again, grow really well. This is essentially a dry farm up in northern Washington. It did really, really well. And we're very excited to see how this plant can perform across the state. This is another grower partner down near us, near Prosser and Sunnyside. The variety here that looks yellow, it's supposed to be yellow. It's a variety called Fibroir. It's a French variety. It doesn't look green when it's mature. It kind of likes to look yellow. One of the things that's really exciting about this variety is that it requires significantly less nitrogen than most other crops, and especially other hemp varieties. If you give it 100 pounds per acre, it actually doesn't like it. It likes it more like the 50 to 30 pounds per acre nitrogen in the soil. And so it does exceptionally well and produces very high quality fiber when you grow it that way. You can get it to be a little bit more robust and grow a little bit bigger if you give it more nitrogen, but you actually don't get an increase in fiber, and the quality of the fiber actually goes down. So it's important to understand what your soil is like, what your nutrient quality is like in the soil, and what the requirements of the individual varieties are. And then when this gets mature, this is the Futura 83 again with a couple of my students and me out there in the field. This is in the same plot in the kind of in the background of that big picture there. And again, across the state, these varieties do very, very well. And we're very excited to work with our grower partners and the seed suppliers to help us evaluate and determine which are really going to be most suitable for industry development in our area. Back to the essential oil evaluation. Here are the students and I out in the field. We're doing sampling. We're looking actually here at terpene production and cannabinoid production over time. We've collected samples from marked plants. You can see the little flag there. So we can go back and know where they are. This is early, about halfway through the season. Later, the plants are taller than Drew and Toby there and me. They grew really well again. And we're very interested to see how the results are going to turn out for the cannabinoid and terpene development. They're still processing all of the plants and extracting them, and we're going to be analyzing those very, very soon for the chemical profiling. All right, but as we're doing this research, we do also have some issues that come up along the way. And some of these are biotic and some of them are abiotic in nature. One of the things that has affected the Western United States, especially over the last several years, has been wildfires. Although I understand New York and the Northeast was affected by this last year as well from these big fires that were up in Quebec, if I remember right. You also got to deal with the smoke. We've been dealing with it for a number of years in the West. 2020 was an exceptionally bad year. We had fires all over Washington, all over Oregon. Some days were so bad, they basically said, don't even think about going outside. The air quality was horrendous. You could see maybe 20 feet in front of you as you're walking outside in some places. It was really, really awful. What does this mean for our crops? Well, we know that if you are a wine producer and smoke is in the area, some of the compounds, and they've actually finally figured this out. They can go look this up if you want. There's a process called smoke taint, where compounds from the smoke are taken up by the berries, by the grape berries, and then they are sequestered in the vacuole of the berries so that they are no longer available in terms of human easy access. So if you taste the berries, you go out there, your field has been exposed to smoke, and you go out there and you taste the berries, they actually don't taste like smoke. They taste fine. But what the plant did is it took those smoke compounds, which are actually quite potent in aroma and flavor to humans once they're released, and they glycosylate them and sequester them in the vacuole of the grape berry, so you can't taste them. But you put those berry in the winemaking process, and all of a sudden that glycosid, there's glycosidases there, and that glycosidic bond is broken. And when you open up your wine after it's been sitting for months, all of a sudden you wonder, why does it smell like smoke? Why is it so bad? And that's what they call smoke taint. It's a real problem for the wine industry. Now that we know what those compounds are, they can actually test for them. There's a possibility that hemp is going to be affected the same way. So if you have a fire like this, putting smoke in the air, you end up with smoke in the air where you're growing your hemp. This was an autoflower variety that was grown by a grower partner back in 2020. Autoflowers, this is about the best you can get for autoflowers in Washington. I think this is the best field anybody's ever had. Plants get to be 1.5 feet tall. That's it. I know in the South they grow a lot better, but in our area they really don't grow well. And the smoke taint really, we think, affected them as well. Also, as you can see, sun does not penetrate to the plants very well. It affects the growth, it affects the yield. Lots of things can be affected when smoke is present. So this is an issue. Luckily, 2023 was much better. We had much less smoke in our area. You guys on the East Coast got to get it. We're able to benefit from the lovely benefits of smoke instead. We're happy to pass that along for the future, but we don't know what's going to happen in 2024. This year is a new year. We could see smoke come back. We could see the wildfires come back. So this is going to be something that's a concern going forward. We have to figure out how to deal with it. And there's reports here, that basically the hemp industry is being very severely affected. And this is there was a nice story about this. It's really pretty sad. That same year, we also had a major windstorm come through, knocked over a bunch of plants, especially taller plants that people had. Some individuals were looking at hybrids between fiber and essential oil types to see if they could do a dual crop there. And a lot of those plants were pretty severely affected. As you can see from the pictures here, they were knocked over. Eventually, most of these fell on their side. They had to go out and harvest them much earlier than they were hoping to before they rotted in the field. It was a pretty bad situation. So lots of environmental effects can impact your crop. You can also have something like a heat wave in 2021. So the year after the major smoke, we had a major heat wave that lasted for 2 years. 118 Fahrenheit for those that are not in the US, that's about 48 degrees centigrade. It's really hot. That was a high temperature for 2 weeks. It never got below 90 Fahrenheit or about 32 centigrade at night. The plants did not like it. Unfortunately, this hit unexpectedly, like less than a week after we put our plants in the ground. We were lucky we were able to keep water on them. They came up. They look pretty chlorotic. They don't look that great, as you can see. And then you see there's lots of weeds that came up here too, the lamb's quarters and the pigweed, which are major weeds in our area. They were not as affected. They came up actually after the heat wave. And we had a problem that whole year in dealing with this weed problem. We didn't have at the time chemical control for weeds available to us. So we had major impacts on the crop that we saw. As the year went along, we still, major weeds came in, kind of really outcompeted the hemp. You really need to get canopy closure by the hemp. If you get canopy closure, nothing outcompetes it at all. But if you don't, you can have serious problems with these weeds. This is like the best looking plot that year. It looks pretty bad. These plants got to be, this is a Carmagnola variety from Italy. These plants got to be 12 feet tall, like we kind of anticipated, but it's a handful. This should have been a very dense plot like you saw on the previous slide. But it didn't because the weed problem was pretty severe. Dealing with weeds is a big issue. Finding weed control methods is a big issue. We think that planting at the right time for your region, getting the crop up, getting canopy closure before the weeds come in is really critical. And experiments we're doing this year are going to address this question as well. And if you look at the essential oil hemp varieties, they're in the middle, what looks like it's a little bit darker green. These are our hemp plants trying to compete against the grasses. We had to do so much manual weeding this year. It was awful. Trying to keep up with it was a big challenge. So again, but eventually they did do better, and they kind of look like this. They don't look great. These are the ones that on the right here are plants that are under plastic. You can't see the plastic because the plants grew so well. But the plants on the bare soil, we were trying to do a higher density, and they just really did not perform well against the weeds. The other thing that happened with that heat wave, some of the varieties that we had planted really were affected. You can see the ones here on the left or in the background, they actually look pretty good. They had good yields. But this particular variety in the middle here, it did not. The roots were affected severely. The plants grew. They actually looked really good early on compared to other varieties, but then they stopped growing, and it looks like the roots basically just kind of rotted and they fell over and didn't do well. The next year in 2022, they did a lot better. We grew the same variety again. There wasn't a heat wave. They grew a lot better. But when there was this major heat stress, this variety did not perform very well. And the other thing that we saw was some biotic effects. We have insects that come in, and we have some viruses. Beet curly top, this is an example of what it can look like on a hemp plant. It's spread by leafhoppers. And it is endemic to Washington State. That's actually where it's from, we think, originally. It's from our part of the world. It's spread across the United States from here. The leafhoppers live on 200 plus species in Washington State. All of those can contain the virus. It was originally identified, obviously, in beets, but it's found in many different crops. It's in beans, it's in carrots, it's in onion, it's in most crops. It doesn't really have much of an impact. Most hemp plants, it doesn't have much of an impact. Here's an example of one where it does. And some varieties were much more susceptible than others to this. Some varieties appeared to have no impact at all from this virus. We did some testing. We evaluated plants to see if it was present. Almost every plant had the virus. Almost every plant that we tested had this hop latent viroid. Most of the plants looked beautiful, but a few of them didn't. So there's definitely a genetic resistance effect occurring here, and trying to figure out what that is going to be really interesting, I think, as well. But there's been a lot of interest and concern in especially the indoor cannabis industry about this hop latent viroid, especially in areas like California. As I said, we see it in every single plant that we looked at, and most of the plants don't appear to be affected at all. Now, maybe there's a 10-20% yield decrease that we are not even able to tell because it's there in everything. And if we were to get rid of it, we would see it actually a better performance of the plants in our area. That could happen, but there's actually no way to test that. This viroid is everywhere. Our fields are surrounded by hops in our area, and it's just going to be in every plant that we deal with. I guess an advantage there is that we have a possibility to screen or grow varieties that are more resistant to it. Those that perform the best are potentially going to be more resistant to that viroid. What we do see as the most interesting impact, though, is that if you combine beet curly top and hop latent viroid and another virus, when those three are present in the plant, you see symptoms like this, only worse. And so it looks like if you really hit the plant with a multiple pathogen attack, it has a hard time resisting that. If one of them comes along, it does pretty okay. But if lots of them are there, if they're all attacking it simultaneously, then the plant could be severely affected, we think. We need to follow up more research to figure out if that's really the case, but that's our current hypothesis at this time. Okay, so now I'm going to talk briefly about our efforts in the National Germplasm Collection. Again, this is in collaboration with Zach and Gordon there at the Geneva ARS facility, but also the project is being led by Charlie Brummer and his postdoc Daniel Papp at UC Davis. Here's what our field looked like back in 2022. We have a similar setup last year. I don't have as good of a picture from last year as I do from 2022. I got a better image here, so I'm showing that one, but it looked very similar in 2023. And what we're doing is evaluating numerous accessions in this germplasm collection, about 40, 35 to 45 each year, and seeing how they grow individually. We're looking at their growth, performance, and we're looking at seed quality and a bunch of different factors. As you may or may not know, in the United States, we have this germplasm system, National Plant Germplasm System, as it's called, NPGS. And there are different facilities around the country. One of those is in Geneva, New York. One of those is actually in Pullman, Washington, where I'm located. In Pullman, the collection for beans, like broad beans, dry beans, like pintos or kidney beans or black beans, the national collection for beans is here, and for many grasses is here, and for some other crops. For hemp, however, the national collection has been set up in Geneva, Washington, and that's where Zach and Tyler are, and so we're working with them. These other sites have other crops where they focus on maintaining the germplasm. If you are interested in accessing any crop and getting seed from any crop, you can contact this system and you can request those seeds and they will ship them to you for free. Now, they don't give you, bucket loads of this. They won't give you a freight train car full of it, but they'll give you about 100 seeds that you can work with from any given variety, any accession that you're interested in. And so you can use that to evaluate different crops for your area or even for breeding efforts as well. So we think this is a wonderful system that's available to everybody in the world, not just the United States. And I try to emphasize that. Many people in the US know about it, are using it, and are finding that it's very, very beneficial to them. In hemp, and in fact, the broader plant, Cannabis sativa, as you, I hope, know, Cannabis sativa is one species. The high THC versions are used for quote unquote marijuana or high THC cannabis or cannabis. There's different words that are used for it now, for medicinal application or for recreational purposes in different parts of the world. And then there's the industrial hemp versions or the chemical hemp versions that are used for other applications. It's all the same species. And there are just different versions of it that have different properties that have been bred over the generations for thousands and thousands of years to have these different properties. And those are now spread across the world. And this germplasm collection is working on collecting individuals from those different populations around the world so that we get the best quality diversity into our collection, so it's available for all these efforts that we have for improving the crop to support the industry development. Ways that we look at this is, again, we collect and conserve different genetic resources. These are then distributed. Part of this, of course, is you need to know what it is that you're looking at. So phenotyping and genotyping the material is very, very important. And we also, Zach and Tyler especially, are working on developing tools and methods that will help serve the hemp community for the ability to look at these plants and use them. This germplasm collection has great value, I believe, I think many other people do, to help support the industry because it provides novel phenomic and genomic resources and scientific tools for hemp. It's helping to develop those. We get good information about diverse traits that are being mapped onto populations. This then leads to genetic material and diversity that can be integrated into breeding programs. They are developing some pre-breeding populations, which will then be used in future efforts to do larger scale breeding efforts. And this helps us, we think, in dealing with issues such as germplasm bottlenecks, because we're able to get the biggest diversity of germplasm into the system and into these breeding programs. The way that this has gone over the last several years is we're now at close to 600 accessions that have been acquired. There used to be a collection a long time ago, but as you may know, in the early 70s, the world started this thing called the war on drugs. Cannabis was particularly targeted, and the germplasm collection that was in existence at that time disappeared. There's some information about it, but none of the material actually existed. So we started from scratch back in 2021 to rebuild this collection. Zach and Tyler leading the effort, and those of us at partner institutions helping them. we're doing a lot with the phenotyping, which I'll show in just a second, to help develop this system so that those interested in using this germplasm collection will have interesting and useful information available to them. The trials that are being conducted for the phenotyping effort are around the country. We're up here in Oregon, above Oregon in Washington. Our initial effort was in Othello, but we've now moved to Prosser. We have a better facility there. We got actually permission to grow there finally. But there's facilities in California, Oregon, New York, and down in Louisiana and here in Tennessee as well. We're looking at traits such as flowering time, sex determination, plant architecture, fiber quality, grain quality, such as yield, protein content, fatty acid content, and profile and diversity, and specialized metabolites such as terpenes and cannabinoids. And we're also very interested in this G by E connection, genetics by environment. If we grow the same things in these different regions, which are very different from each other across the nation, what do we see in terms of the results? Very interested to see how that all pans out over the years. This is the pipeline, how it works. Geneva is the location, central location for all of the information. The collection is maintained there. They have database, this Green Global. If you look this up, you search Green Global, you'll be able to find the accession and the accessions that are available and request those. That provides information to breeders and researchers. And then we're right here in this gray one, kind of in the middle, as part of the phenotyping trials. Others are involved in other parts of evaluation of the products and also maintaining the germplasm collection. But we're very excited to be part of this. It's very interesting to see how these different varieties and different accessions perform and grow in our area. Part of how we're doing this is we're looking at growth characteristics using what we call a kite kind of a diagram, where you look at the diameter of the stem, you look at how tall it is, you look at how wide it is and how tall it is to the width. Different accessions will have different shapes of this kite. Some will look like this, some will be inverted, some almost look like an upside down triangle, depending on the variety. And those are very interesting characteristics that can tell us information about what that variety might be potentially used for, whether for fiber or for some other application for grain, or if you're growing it for CBD or other chemicals, the architecture of the plant can have an impact on productivity and yield. And so understanding the growth characteristics of the plant is very, very important. Look at leaf phenotype as well, see how big the leaves are, what their structure is. We look at flowering date, days to flowering, and we see big differences depending on the variety. If we look at cannabinoid level, again, we see very big differences in types of cannabinoids for the different accessions. Looking at CBD, CBCV, et cetera, you see a list here. All of these, if they are hemp, they have very low levels of THC. There is the ability in the USDA germplasm system to maintain some varieties that end up having higher levels of THC. We don't actually grow those at our facility if we know that they're high, because we have to work under the USDA based hemp programs, which means we have to grow hemp that's lower than 0.3% THC. So those are the varieties that we're evaluating, although the collection itself has had some donations that are higher. And then what we are doing in my lab is we are doing a lot of the phenotyping for the seed protein as well as lipids. We also can do the chemical analysis too, although our partners at the ARS are doing that for this project. But we can look at cannabinoids and terpenes, et cetera. And some examples of results that we see from these efforts are really interesting. For example, we had a CBG cultivar that we were looking at, one of our grower partners grew. We took individual plants from across the field. They all looked really the same. If you look at them morphologically, it looked like what you would call a pretty good variety. Some of these varieties are highly variable in morphology. I would not call them varieties. I would call them populations, although sometimes they're listed as a variety or a cultivar. This was one that looked like a really good cultivar, but the chemistry was very different when we compared individual plants to each other for CBD, CBG, and even THC. And we see this repeatedly across a lot of these accessions and varieties and cultivars that we look at, that there is a lot of chemical diversity that is maintained within these different populations. To us, that suggests they're not at the point where I would call them like a true breeding line. But that might actually be very important and useful if you're growing them in a field with a highly variable environment where you've got pathogens and pests coming along that could be deterred by different chemistries. So in some ways, having a diversity of chemicals is important potentially for the crop to be successful, But if you're growing them specifically for certain compounds like CBG, you really don't want a lot of plants in your field that have THC and CBD, right? So information like this is important as we move forward with breeding efforts. And we see the same kind of thing with terpenes. These are some of the major terpenes that you typically see, but we see a big difference in the ratios to each other. Again, this is a cross that was made. The same exact type of genetics was what was claimed, but the chemistry, again, was quite different. If we look at the seed oil content, we also see a big diversity across lines. These are all grown for fiber or seed or dual crop purposes. And we see that some of these, if you notice, some of them have like an abbreviation like this Bieler-Brzezinski. We got seed from a couple of different sources, and we saw differences in the content of the oil. They were grown in the same location, same field, but the seed source gave us a slightly different result. For this X59, we really didn't see much of a difference. Some places see X59 oil content a lot higher. We don't. It's pretty much average compared to the other ones that we've seen. For some of our other varieties, like this Jinma, this is a Chinese variety. It's really only grown for fiber. It has very large seeds in size, but very low weight seeds. They actually tend to be mostly hollow. They have very low protein, very low oil content. And I would definitely not grow this for seed. People definitely grow it for fiber instead, but it's interesting to use those as a comparison. We also looked at not just the oil content, we looked at the fatty acid composition, and we saw that there was a pretty common trend, which is that the polyunsaturated fatty acids here in purple and black and this tan or peachy color, that's the majority of the fatty acids that are present in hemp seed. You throw on the monounsaturated C18-1, you're like typically 90% of the fatty acids in the hemp seed oil are mono and polyunsaturated. Very different from most other crops. It's one of the reasons why, number one, it's great for your heart. This has omega-6 and omega-3 fatty acids in it, which are very interesting, but also for industrial applications. Because of the highly unsaturated nature compared to other fatty acid bearing crops, it has great potential for certain industrial applications like in making glues and resins, etc. So finding out which variety differs from others is also very important here. And one of these, X59, did have a big increase in this omega-6. Of course, the omega-3s are the ones that you're most interested in for your heart. So that's something to think about. And again, here's, as I said, different ones, the same variety. We did see this, though, is that even though we saw a difference in yield, as I showed on the previous one, the composition really did not change much. So what we think is happening here is that the composition of the fatty acid is probably most significantly controlled by the genetics, and the yield is affected, and sometimes substantially, by the environment. And that environment could be the current field that it's in, or it also could be an epigenetic effect where the parent, where the mother plant, where the seed came from, was grown. So if you get seed sources from different locations, you could see a difference in your fatty acid yield if you grow it in the same exact field. We're going to follow this up with seeing what our results from last year tell us if we see the same kind of result. This is an interesting preliminary trend, though, suggesting this might be something really interesting for the industry development down the road. Okay, and that was the Jinmo one. So conclusions here for the phenotyping. The hemp varieties are often highly variable, and I put varieties in quotations there, and they appear to be populations instead of really true breeding lines. The seed composition is genetically determined mostly, we think, but content can be significantly impacted. And these biotic and abiotic stressors, such as a heat wave or smoke or a virus, can have a significant impact both on our phenotyping efforts as well as potentially on the plants themselves. So what does all this mean for industry development in the next couple of minutes here? Understanding how to grow the plant, having the production requirements figured out for your area is really critical. We know now what we need for how to grow the seed, what kind of soil preparation is needed, and understanding which varieties to select for your evaluation and ultimate production is also critical. These are some different fields grown in 2019 by some of our partner growers, and you can see they're very, very different from each other. This is supposedly, this is the same crop, it's all hemp, but it can be grown very, very differently depending on where you grow it and what your purposes are. And those different growth parameters are really important for what your ultimate uses are going to be. So you need to think about that. The other thing you have to think about is how you're going to harvest it. Yields for the plants for CBD can be as much as 1,000 to 2,000 pounds per acre for flower varieties. Fiber, these plants can easily be 10 to 18 feet tall or taller. You can yield 1.5 to 5 tons of fiber per acre, depending on where you grow it. Grain yield can be 1,000 to 1,600 pounds per acre. A lot of places it's lower. It's in the mid 400 to 600 range. In our area, they tend to be in this higher range. If you're going to grow it for a cut flower market, you have to do it by hand. If you're going to grow it for seed, using a combine is what you want. If you're going to grow it for fiber, you're going to want something like a sickle mower. Grain and fiber together, you're going to combine it and then do something like a sickle mower. If you're growing for super high quality fiber applications like textiles, you're probably going to want to have a retting system involved. A lot of this is going to be helped by automation, by equipment. A lot of that does not exist in our area. It is being acquired and developed across the nation. And we're going to see that trend growing. And over the next few years, we're going to see a lot more availability of equipment that will help us with the harvesting efforts. And then finally, you got to ask yourself, well, do I really want to grow this? Is it something worth pursuing? Well, the first question, of course, if you're a grower, is you have to ask your question about profitability. What kind of yield am I going to get? What kind of price am I going to get? And then what are my input costs? If I'm growing it, is it going to replace something on my farm or in our area? We think that it actually adds benefit by being a rotation crop. And that's what that new grant from DOE is going to help us support investigating, is looking at its integration into crop rotation systems. What does it do for the impact on other crops? There's been some suggestion and some papers published on this, that in wheat especially, but also maybe other grass crops like corn, you can see a bump in yield in that grass crop if you grow it after you grow your hemp. And we think that a reason for that is that the hemp does some very positive things to the soil, loosens it and makes nutrients more available to other crops after the fact. You do have to deal with government restrictions and red tape. For example, you have to put GPS coordinates of your field, you have to test for THC, et cetera, but those are not really that onerous now that we've figured out how to make this work. You do also have to consider things like risk. What happens if you have fires in your area? What's that going to do to your yield? What happens if you have windstorms? What happens with whatever else that might happen? Is there crop insurance available? Well, when I first put this slide together back at the beginning of 2020, there was no such thing as crop insurance for hemp. That has changed. That is now becoming available across the nation. But are there contracts available for you to grow it and to be able to produce it? That, it kind of depends on the area and the year. Where, of course, most growers are going to demand this if they're going to grow this as a crop. There's a big risk involved potentially of growing this, and they want to know that they're going to be able to get something out of it at the end of the day. They don't want to grow it and then have bales of fiber sitting on their field because somebody doesn't show up to pick it up because they don't have a contract. The market, what that's really going to be in your area is really important, whether it's going to be fiber, seed, or CBD type hemp. And again, does it displace something in the market? We think it actually augments the market. I think if it's going to displace anything, we're kind of targeting petroleum. We don't see this as displacing other crops. We think it as supplementing other crops. And I was at the recent multi-state meeting again for the S1084 down in North Carolina just last week. And we got to go tour a facility in the area there that's doing some really great things for fiber production. They work on cotton and hemp, and they're showing how you can integrate those together to yield some of the best quality products. So that's really exciting to see that kind of thing develop. And we're going to see more of that in the coming years, I'm sure. How big is the market? That's still an open question for everybody, but the market potential is actually huge, a lot of people have said. Now, if you look at actual costs, this is what I get for the most recent data as of yesterday for corn, soybean, and wheat. These are, if you're going to grow an acre, this is what you might expect if you have a high yielding, a very, very good crop, depending on where you are in the country. This is what the price is, and this is what you can expect per acre to get as revenue. If you're growing hemp, these are the typical prices the last 2 years. For floral hemp, we're actually up to about $3.50 a pound again. It was down to less than $2 2 years ago, but it has rebounded. That oversupply has been consumed, and there's actually a demand again for this. And we're back around the $3.50, $4 a pound. If you have these price points, all of a sudden this becomes quite attractive from the profit perspective. Now the question is, profit includes revenue versus your input. So you have to consider what your inputs are. What is it going to take to grow the crop? This is what you could get for your gross revenue, but you still have to consider what your inputs are going to be. And that's going to depend on the particular area and even the particular farm. So one of the ways that we're also trying to help with this is looking at this broad picture. Again, Oregon State University with Jeff Steiner as a lead put together this Sustainable Ag Systems grant where we're looking at how do we set up a bioeconomy based on hemp in the West Coast. And we're very excited to see how this is moving forward and information that we're generating that will lead to some answers to some of those questions. I'm going to skip because we're out of time. Just highlight here again, there's many opportunities here and some challenges, all looking at varieties available, pesticide issues, harvest equipment, manufacturing, and really what are the market opportunities? If we have great markets, great opportunities, all of these things as potential issues go away because somebody will find a way to resolve them. So with that, I'm going to close with the acknowledgements. A lot of people are involved in these projects. Very excited to be involved with them. And I'm going to turn to questions at this point. We have a couple of them, I see. So if I understand right, Bahia, I should hold these up. Okay, how old were the seedlings when transplanted? We typically transplant them between 21 and 25 days old. We don't like them to grow longer than 28 days. They tend to get too big. The big issue is we want to avoid seeing the roots get bound up in the seedling pot. That's a really critical issue. You have to make sure that they don't get root bound. And as long as they're not root bound, they seem to do okay. But yeah, that's our typical age. We've tried planting them earlier when they were even younger. It's a little bit more difficult with our equipment to do so. We did do that for the SAC grant. We put them in these, basically it's a peat cup that we put soil in. Again, we see the same issue with the roots. You don't see a taproot, you see branching roots instead. I think having to go through that cup is an issue. So direct seeding seems to eliminate some of these problems with, I don't know if it's a problem, it's just a difference in how they grow, right? So, and again, it may depend on what you're targeting and what your ultimate end goals are. At the same time, the roots branching, they tend to then focus on the upper 12 to 18 inches of the surface of the soil where the root zone is. So you need to consider your watering, how your water availability, if that's the case. If you're irrigating, it's probably not much of a deal, but if you're relying on groundwater, then that might be an issue in terms of how well the plants will perform. Did you have any issues with seed quality? I'm not sure exactly what issues that might be, what you mean there. The results, I mean, if you're talking about the results from the varieties, low germination and moldy. Okay, that's a good question. Thank you for following up. We have not had a problem with moldy seeds at all in our area. Everything that we got has been very high quality. The low germination we have seen with some of the germplasm collection accessions. Some of those are collected from feral populations around the nation, which basically it's weed growing as a weed, if you want to laugh at that. And as a result, for those populations to survive long-term, having a seed bank in the soil is important. The varieties that are grown purposefully for production, they typically have a germination rate exceeding 85%. Everybody claims that. We typically see they're in the 90s, 95. Some of them are almost 99 or 100%. We, out of a thousand seeds, you see maybe one that doesn't germinate. Some of those other accessions, 3% germination is what we saw in one of them. It was really, really low. It depends on what the source is and what exactly varieties or accessions you're talking about. And for protein quantification, we actually have, we're developing a new method for that. The way that we have done this in the past is kind of the standard. You measure carbon and nitrogen using combustion, using a standard lab, then you multiply that times a factor that supposedly tells you accurately what your protein is, even though it's, that factor is the same for every single seed across every single species. I don't think so, but that's what everybody does. We are developing instead a method based on a much smaller scale that we can use one seed for. And so we can get actually the protein content in an individual seed. It's using more standard, what I would call biochemical based approaches. And students in my lab are optimizing that right now, working with Phil Bates, our partner here at WSU, who does lipid analysis. And they've developed this method for other oil seeds, and we're adapting it for hemp right now. We hopefully will have that available within the next couple of months up and running. And then by later this year, we hope to have our first paper out where we show data for the seed yield for these different crops, and the method will be in that as well. Next question, is there any data of varieties of higher terpene diversity. In our effort, we are kind of just looking at that right now. So I can't answer that question yet. Hopefully within the next year or so, we'll have some information. But there is a big diversity if you just look in the literature from what people have found. But in terms of if there's a higher diversity within varieties, I don't know the answer to that. And what do I think the best hemp types are for Washington? All of them is the short answer. CBD hemp grows exceptionally well here. You know, Washington and Oregon were the hub of the black market marijuana industry for decades. Varieties were grown and developed here for that. All you got to do is knock out the THC, which people have done, and those same varieties perform excellently here in the area. We get great yields. The day length is perfect for most of these. We're in the northern latitude. We're even better than Oregon in a lot of cases. We get higher yields. And the quality is very, very good for CBD type hemp or CBG or CBC or CBDV, all of these other varieties that people are developing in our area. Fiber grows exceptionally well as well, and the grain varieties do well. The grain we think is going to be really good for our dryland areas. Near where I grow, where I currently live in Pullman, we have the Palouse. We get 22 to 24 inches of rain each year at the right time of year to help support hemp. We have a couple of growers that have grown here, and they've got really, really good results using dryland farming. The central part of the state, it's kind of a desert. Rain is a lot lower levels. It ranges between 4 to 11 inches in a lot of places. But we have the Columbia River that runs through, and they have high availability of irrigation water. So, you know, if you want to use irrigation, you can grow hemp really, really well in the central part of the state. If you want to grow it without irrigation, the northern and eastern and western parts of the state are very suited for that. So, and do most fiber folk in Washington irrigate their entire fields? Well, the answer is that there's not very many fiber folk in Washington, to be honest with you. We've kind of been leading that effort with our partners. And yes, in the central part, we do irrigate because I just answered why, because there's not enough rain. But on the eastern part of the state, we don't have to irrigate, and they don't irrigate at all. In fact, there's no irrigation availability in eastern Washington. We don't have any kind of river water supply in the area. We really rely on snow pack charging the soil and then rain that comes at the right time of year. I think that's all of the questions that we have, unless anybody has another one. I can stick on for another minute, I think. We went over a little bit. Thank you for those that stuck with it. I hope this was interesting.
Yeah, thank you very much, David.
This has been a production of Cornell University, on the web at cornell.edu.